Okay, so next we're going to talk about what's been simmering in the room over the last couple days since we just heard some of the plans for both Ionis and Ultragenyx as it relates to their upcoming phase 3 clinical trials, which we're going to hear a lot more about from them in the later sessions. But we asked Jennifer if she would really speak to us about what controlled studies look like. It's very important for our community to understand what that entails, what that looks like. There's a lot of misconceptions of what that is. And so we thought it would be a good idea to really explain and understand what controlled clinical trials look like. So Jennifer's going to talk to us about that. As you well know, Jennifer leads regulatory and compliance for FAST, and she also is the Chief Operating Officer of AS2 Bio. So she is a force to be reckoned with. She's leading the charge. She has probably more incredibly precise drug development experience for rare disease than anyone that I've met in the last 10 years, and I would say that that is a lot of people because I talk to a lot of companies as it relates to these types of therapies for rare, not just Angelman. So we are so lucky to have her, so please give her a very warm welcome. Jennifer.
Thank you so much for that very kind introduction. I'm really excited to be here today to talk to you about controls, and I'm just going to give you some basic background information because you're at a really exciting time where trials are transitioning from early stage into late stage, and they may look a little bit different than what you've seen before, so we thought it warranted some background, and I think this will help you when you see some of the presentations later this morning that will go into trial design late stage. So Allison covered my disclosures, and I'll also say that sometimes I help other companies that have non-Angelman programs, and I still have a retained interest in genetics. So I'm going to also start with a disclaimer, which is, based on my experience in industry, this is just my 2 cents. There are always exceptions to the rule, but I'm going to give you sort of the general view of how things work. And I'm not commenting on anyone's specific design, I'm not giving you advice, I'm just giving you the basics. So let's start at the top about studies. And generally there are 2 types of clinical studies. There are observational studies where subjects are just followed over time. And an example of that is the natural history study that you heard Anjali talk about yesterday, where they're just kind of watching how symptoms behave over time and learning more about the disorder, but they're not trying to give you an intervention. So interventional studies are ones where a participant is assigned to a group where they try a drug or a device. And that's what we talk about when we mean clinical trials, are interventional studies that are evaluating maybe a new investigational therapy. Trials have phases. Usually they start at phase 1. And that's a small number of subjects are enrolled. And really at this stage, they're just kind of looking at safety, just making sure they understand how to dose it. They might look at different dose levels. And most importantly, that there are no concerning risks. If that goes well, you progress to phase 2 and more subjects are enrolled. And now they're looking more at the safety, maybe how to optimize the dose, and does it work? Is it improving symptoms? And then they're learning and thinking about how to design a late stage study, phase 3, where now what you're really trying to do is just confirm definitively that the drug is safe at the dose that they chose and that it's effective and that it meaningfully changes symptoms that are important to caregivers. But in rare disease, it's different. You have less patients to study, and oftentimes you're trying to learn quickly and move through the development path very expeditiously because you want to get product to patients if it works, because obviously there are serious unmet medical needs and patients are waiting. And so what you see is these phases get combined, where you might have a phase 1-2, and then a 2-3, or just a phase 3, where they just go early, look at safety, and then move into a definitive study that's really meant to establish that it works, and you compile all that information, and hopefully you file it to the FDA and get the drug approved. So in early stage studies, these are often not controlled, meaning there's just one group, everybody gets the active drug, you know what you're on, the doctor and the participant knows what they're receiving. That's called open label. And that's what you've seen a lot of. So a lot of the trials that have been ongoing over the past several years, oligonucleotides, those ASO programs, the Neuron program, these are open label. Everybody is getting the active drug. But in late stage, now you move into a controlled experiment because they're really trying to provide strong evidence that the drug is safe and that it is effective. And that's why a control gets introduced. So now let's talk about controls. We'll start on the left. There's the placebo. I'm sure you've all heard about placebo-controlled trials. This is really a dummy pill or maybe a sugar pill. And so whatever the active drug is, the investigational drug that's being studied, if it's a pill, if it's an injection, if it's like an oral solution, there's an active and then a dummy that's meant to look just like it. It looks exactly the same. It tastes the same. It's meant to make you think that you're getting the active. And so you don't know if you're actually taking the drug or you're taking the placebo. Then there's active controlled trials, and this is when you have a drug that's approved. So imagine you're studying a drug for blood pressure, and there's lots of approved drugs for that, and they're very effective. So you don't want to take a patient off of something that's helping them, but you want to try a new drug, and so you might study the active approved drug versus the new investigational drug, and that's an active control. We don't have that yet for Angelman syndrome, but I'm hopeful there will be a day where we have enough effective therapies that we need active controlled trials. Then there's sham. A sham is a procedure that might be sort of invasive. It really depends on the modality. But one group gets the active and another group gets a pretend procedure that's meant to make them think that they got the drug. And so you see this sometimes in gene therapy trials. There's trials of Parkinson's disease where maybe there's an invasive procedure to inject the drug into the brain. And so you don't really want to make a patient that's not getting the drug go through that whole procedure, but you still want them to think that they have so that you have some objectivity in the study design. And so one group gets a pretend procedure. And then there's historically controlled trials. And this is where there's only really one group in the study and everybody's getting the active drug, but they compare those results to an external group of patients that never got treatment. And so if you think about the natural history study, you might see, and sometimes this happens with gene therapy studies, where there's a group that gets active and then they get compared to the natural history study to really understand if there's differences in treatment versus what happens when you don't treat a patient. The challenge with historical controlled trials is that the FDA and some of the other regulatory agencies don't really love them. They allow them when the situation warrants it, but they sometimes have questions about how alike the two groups are. Is the historical group really the same as the treated group? Do they have all of the same care? And so sometimes they worry that it's not a great comparator. So that's why you see controls even in rare disease studies. Generally when you're in a control group, generally when you're in a controlled study, you're assigned at random. So you have a random chance of getting in the active group or in the control group. And when there are controls, generally studies are blinded. And the double blind is when neither the participant or the doctor knows what group you've been assigned to, and it stays that way until the study is over, and you break the blind. And then usually there's a period of analysis before you find out what drug you were on. One note about sham-controlled trials is that generally there is somebody at the site that knows what drug you are getting, and the reason why is because they have to perform the procedure. So you have a doctor maybe at the site that administers the procedure, and this happened with spinal muscular atrophy. So there was a study using a lumbar puncture in very young babies, and there was a concern about making a very young infant go through that lumbar puncture. And so one group in the study received the lumbar puncture with the drug, and the other group basically had a pretend procedure where there was a small pinprick made in the back and a bandage put over it to make the family think that the child had received the study drug. And when this happens, somebody has to know what they're on. But it doesn't mean everybody does. The rest of the staff at the site, so maybe the main study doctor and certainly the participant and their family, don't know what they're getting. So this is a difference here. Somebody has to know in that situation. But companies that use these sham procedures go through extensive processes to set up blinding so that nobody finds out what their assignment was. So they take care in making sure the people that know and the people that don't know aren't talking about it. So then the question is, well, why do we even need these controls? What is the purpose? And generally, it's the gold standard in clinical research, right? It's sort of, a well-designed experiment. You randomly assign, you blind so people don't know. So you don't introduce bias into the study. And that means that your attitudes towards the drug aren't reflected in the outcomes. And so you can think about a drug, maybe you get into a trial and you're really hopeful about it. And so you start thinking that the effects are coming from the drug when really they're not. And so The control group helps you sort out the differences. They're also useful to compare outcomes when the disease is different. And so when you have variability like in Angelman syndrome, it helps you weed out what is really just a feature of development versus what's really an improvement from the drug. And if you think about some of the information we heard yesterday from the natural history study, you can see that even in Angelman syndrome, there's a little window of development. And if you think about walking ability, right? Most children gain the ability to walk, even though it's very late and not on the same time course as neurotypical individuals. But if you were doing a study and you were looking at motor changes, are you catching them in a period where they're just on that small improvement slope, or is it a factor of the drug? And so when you use a control group, it helps you make these comparisons that weed out that noise. It also helps with safety. You know, you get in a study, there's nausea, vomiting, diarrhea. Is it cold and flu season, or is it really a factor of the drug? And the placebo helps you see whether, in fact, certain adverse events can be attributed to the drug. And it really helps definitively establish benefit. So if you're using rating scales or questionnaires, and we know in the natural history study and other studies, there's a lot of questionnaires that parents fill out in Angelman syndrome. And if they're subjective, sometimes it's hard to know really, you know, is there true improvement there? And again, having two groups where one doesn't get the drug and the other one does helps you understand if there's really effects. And I'll give you an example here. If you were in a study where you were evaluating pain and you went in to see the doctor and the doctor said, "How is your pain today?" And you said, "It's not great." How does the doctor rate it? Is it the yellow face or is it the blue face, right? So there's going to be variability in this across the trial. So again, controls help weed that out. Even in rare disease, we use controls, and sometimes it's frustrating, and we think, why do we have to do this? This is a rare disease, and we know, basically how symptoms behave over time. We don't really need controls. But usually the regulators still insist on this because they really want to make sure they understand the true efficacy and safety of a drug. And I'm showing an example here from a drug, Unauthorized, that was approved for spinal muscular atrophy. And they were using a motor function assessment that is assessed by a doctor. And it's good if your score goes up, not down. And here you can see there the blue group got the drug and the gray group got placebo. And at the end of the trial at 12 months, they were different. The treated group went up, the placebo group went down. But if you look earlier, sometimes those lines are closer together and they're kind of like overlapping in a sense. And is this behavior of the disease? Is this, people thinking they're getting better? And so sometimes you don't actually know how the disease is going to behave. And so over time and with extended follow-up and a placebo, you can really understand the differences between groups. And I just want to spend 1 minute talking about bias because it really can impact the results of a study. And I'm showing here an article from the Wall Street Journal many years ago about the impact of social media on clinical trials. And what some groups were seeing is that participants in studies were getting on social media forums and talking about what they thought their assignment was. Like, I'm having diarrhea. I think I'm in the control group, the active group, and not the placebo group because my friend in the other group isn't having this side effect. And so they were trying to really basically unblind themselves to their treatment assignment. And this isn't good for a number of reasons. I'll give you some examples from this report on clinical trials and social media that was written by a medical ethicist. And they give some examples from posts on Facebook where one person was saying, "Hey, I really want to get into this study. I tried this other drug. It really wasn't working for me. And tell me how to get into this other study." And somebody responded and said, "Oh, first thing, if you go in for the study, don't tell them that you were on that drug and you didn't do well because you have to be on it again in this study and you might not get in." And that's not really great because that could affect the outcome of the study. Maybe that drug isn't safe. Maybe it's not beneficial for that patient. Maybe it would affect the results. So we really don't want to sort of bias the outcome by talking on social media about how your experience has been. Another example is one individual posted about side effects, their own experience, and they said, "Oh, there's really no side effects of this drug. I'm doing great. You don't have to worry about it. And if this happens to you, it's not the drug." And that's also not good because it's really important when participants are in a trial, if they're experiencing what they think are side effects, it's really important that you talk to your study doctor about that. That's very important in understanding what the safety of the drug is. And so we really don't want to bias results by talking about them, especially in a controlled and double-blind study. So questions that you want to ask about control groups when you consider studies are really how many patients are going to be in the placebo group and how many are going to be in the active group. Sometimes it's the same number. Sometimes it's twice as many on active than are on placebo. These are questions that you want to understand. How long do I have to stay on the placebo? Is it 6 months? Is it 12 months? Is it 24 months? And then after that time, am I going to be offered active drug? And how soon do I get into that active drug arm of the study? And when will I be told what my assignment was? I mentioned before that even when the blind is broken, there's a period of time where analysis goes on. So it's not as if you find out the day the blind is broken. It might be some period of time. Or sometimes in extension phases, a company might want you to still not know what your prior assignment was while they're continuing to evaluate your effects over time. So these are important things that you have the right to know up front and you can consider. Clinicaltrials.gov is still a good source of information on trials. You can find out about trial designs, trials that are ongoing, but there's also a glossary of terms there if you just want to explore and know more about clinical trials. If you search for trials on Angelman syndrome, you can find the list of studies that are ongoing, those that are finished. But in particular, you can find out about their designs and what their controls are. If you use the study details tab that I'm showing here, it provides information about the design and the control group, and you can dig further to see really, okay, how was it designed and what did they use? Was it a placebo? Was it a sham? Was there no control at all? So these are things that you can do to just dig deeper into some of the things that are out there. And I will stop there and say thank you so much for your participation in clinical trials up till now. This does not happen without you. You are the most important part of this process. Therapeutics will come to Angelman syndrome because of you and your participation. So thank you so much for your contribution and your sacrifice.
We have time for questions. She was very, very quick with her talk. So if you guys want to, well, if you want to keep going, we're early. But if anyone has questions, Jennifer, that was a really excellent talk, and I think it's the topic of a lot of discussion as we enter, for the first time, a true transformative, exciting time in the Angelman community is entering phase 3 with a disease-modifying therapeutic option. With multiple companies headed in that direction. So this has been a real topic of conversation amongst families. So really ask questions. Yeah?
Terry Jo.
Oh, hi. It's Terry Jo Bichelle. I had a question to me at dinner last night, and I thought maybe you could answer it here and it would help more people. One of the moms I sat with, grandmoms I sat with, asked if your child is in a clinical trial and they get sick and go to the doctor, is the doctor able to find out if the child is on study drug or not, and how do they handle that, and do they tell the study if they get sick?
Studies, so first of all, if you're in a trial and you get sick and you go to a different doctor that's not your study doctor, you should still try to communicate back with the study doctor, because that probably represents an adverse event or could, and you want to make sure that the study doctor knows about medical history during the trial. Generally speaking, if somebody has a very serious adverse event and they are concerned about that patient, they might break the blind for that individual patient. That does happen. It doesn't always happen, it's not necessarily the case, but if they wanted to carefully manage the patient in a blinded trial, they can break the blind. That won't unravel the entirety of the study, but sometimes when they're really needing to understand the particulars about patient safety, they can unblind it, but it's the study team that does it, not the patient's normal general practitioner.
Hi, Liz Berry-Kravis from Rush. I just want to really emphasize the point that was just made about social media. We had a trial in Fragile X where the families figured out what the entry criteria for the study on the rating scale and posted it online on Facebook. And what happened there was that we had a drug that actually did work. But because of that posting, people uprated their symptoms in the area where that we were measuring as our key outcome, and then there was a huge placebo effect as people all got better because they had made themselves worse at the beginning of the study. And now that drug, should have been successful, and it wasn't because of this, because the placebo effect was so high. And so it's tremendously important. And people were posting online all their improvements and everything. This is something that can tank your drug approval. And so it's very important in a phase 3 trial not to put stuff on social media, not to talk to other people in the trial about your improvements or your side effects. I cannot emphasize how important this is. These people with Fragile X are still pining for that drug, and they haven't had it for 10 years because of the behavior that went on during the trial. So as a community, I think everybody needs to make a commitment to doing the right thing so that we can get these drugs to everybody, not just the trial participants.
Yeah. That's a great point, and I know it's done with the best of intentions, but it really can unravel the study.
I think there's any other questions.
Hi, thanks for your talk. It was great. So I just have a quick question about you mentioned that you can have phase 2-3 versus a phase 3. So is it purely just a difference in the numbers, or would there be like a particular advantage or disadvantage one versus the other? And what's the consideration that needs to be taken for it to become a 2-3 versus a 3?
So this is getting sort of granular, but sometimes in a 2-3, there's a part of the study where they're still maybe trying to figure out dose, and they're going to evaluate different doses, and then based on that information, they're going to adapt and maybe pick one dose and go into the final phase of the study. So sometimes it's like learning and confirming, and you just do it in one study still. I think here what you're seeing more evolve in Angelman is that we have 1-2s and then just a phase 3 confirmatory definitive study.
And one thing I would also say, just to add to what Liz said real quickly, is that we all live as it relates to Angelman syndrome. I try to not be on social media very much, but as it relates to Angelman syndrome, I think that's where we have our community when we're not here in this room. And so there's a lot of people that have a lot of conversation on social media. We all see it, we all experience it, and we all participate in it. I saw over the last few years people asking a lot of questions about trials, a lot of questions about people's experience in trials, and people crowdsourcing, essentially. And I've also seen this community slap the hand of the people doing it and say, in clinical trials, you're not allowed to do this. We were warned not to do this. We were warned to take these conversations to your doctor and not to other parents in the community because it can hurt the clinical trial. So I want to applaud the parents who were bold enough to jump in on social media and stop the conversation. We always try to as an organization, but many of these are sidebar conversations on personal pages in which we don't have access or visibility into. We always try to stop it. But other parents that are not part of this organization that jump in and try to stop it are doing so good for this community and for these trials and for these companies. So I applaud you guys as well, because that's a hard thing to do when there's a big conversation happening and you jump in and kibosh it. So good for you guys. Continue to do that.